to the second session. Session 2 will highlight dengue fever challenges and innovative solutions. Expect insightful talks on new chemotherapies, vaccine development, novel vector control technologies, and even Wolbachia field implementation. This discussion is led by Professor Eggi Arguni from Universitas Gadjah Mada, Indonesia. Ladies and gentlemen, Professor Eggi Arguni, a lecturer at the Department of Child Health, Faculty of Medicine, Public Health and Nursing Universitas Gadjah Mada, Indonesia. She is also the pediatric consultant at Dr. Garjito Hospital, Yogyakarta, and she is also the leader of Arbovirus Working Group here at the Center for Tropical Medicine, Universitas Gadjah Mada. Her research expertise is about the dengue control, including the Wolbachia implementation program in Indonesia. Ladies and gentlemen, please welcome Professor Eggi Arguni.
Thank you, Mbak Rima, sorry. Thank you. And first of all, so sorry about this voice. Today is better. Yesterday, totally lost. So sorry.
No, it's
But
beautiful.
okay, thank you so much, and appreciate to the committee who invited me to moderate this very important session. And also, we will have a very interesting session in this second session of the World Mosquito Day. And then first, we will have the expert of the vector control in Indonesia. I believe it is also internationally expert. So can I have Pak Triwibowo CV? Okay, for you who had worked with the vector or with the dengue or malaria must be know him very well. He's the Pak Triwibowo Ambar Garjito. He will talk about the vector surveillance. He is the researcher at the Vector-Borne Diseases and Zoonosis Research Group, Center for Public Health and Nutrition Research, Health Research Organization, National Research and Innovation Agency, and also he's the adjunct lecturer at the Department of Tropical Medicine, Faculty of Medicine, Public Health and Nursing, Universitas Gadjah Mada, and also member of the expert Commission for Vector Control and Disease Carrying Animals, Ministry of Health, Indonesia. Without further ado, I would like to offer this platform Zoom to Pak Triwibowo. Over to you, Pak Tri.
Thank you very much, Professor Eggi. Nice to meet you. And today, first of all, I'd like to thank you very much for organizing committee who invited me to present in this very important meeting. Today, I'm going to talk about the dengue vector control. Okay, can you see my slide? Okay, yeah. Good morning, everyone. And before we talk about the dengue vector surveillance in a comprehensive issues, so I'd like to start from the definition of the dengue vector surveillance. So dengue vector surveillance systematically collects and analyzes data to assess vector population, guides, control, and predict the outbreaks. And there are some specific objectives that are very important to know, including the detection of the vector presence, breeding sites, seasonal population trend, and also intervention impacts. So when we talk about the dengue vector surveillance, so I think its association with the life cycle of the dengue vector itself. For example, in this figure, this is life cycle of the Aedes aegypti, and we know that in the Aedes aegypti there are some states in their life cycle, their adult states, and then larvae states, and then also pupae and also eggs. So in every state, it's very important to know and to identify, to predict, and to plan for the appropriate vector control effort. So as we know that the standard dengue vector surveillance methods are consist of the larvae survey. We can use a dipper, funnel trap, or plastic pipette for collecting the larva in the field, and also ovid trap to capture the egg and also immature states, and adult mosquito trapping with aspirator or specialized trap, for example, BG sentinel trap, gravity trap, and also sweep nets, and also spot check at the sentinel or routine sites. So when we talk about the larvae survey, this is a very common survey in part of the dengue vector surveillance because this survey is widely used method for monitoring the dengue vector abundance and providing the direct evidence of the breeding site and vector presence. So actually, the larva survey is very useful for identifying the breeding hotspot and also can be cost effective and straightforward to conduct because of involving the community participation and also routine monitoring. However, there are some limitations that we identified. The larva survey actually have limited use for assessing actual dengue transmission risk, and also labor intensive and time consuming because of requiring access to many water holding container and household. And also the index derived from the larva survey often serves as poor proxies for adult mosquito abundance and potential virus transmission. And we know that larva survey may underestimate or miss vector population when breeding occur in cryptic or inaccessible site. So they cannot directly detect infected adult mosquitoes. Yeah. In this slide, we can see about this some very common breeding habitat of the dengue vector mosquito in surrounding the households. I think we can see look like a cement tank and place for the wudu in a Muslim mosque, and also water dispenser and also wells. So when we use this larva survey approach, so I think we also very familiar with the current parameter for the dengue vector surveillance, especially based on the larvae and pupae survey. We know very well about the Stegomyia index, and that consists of a host index, container index, pre-time index, and also density figure. And in Indonesia, we have also a free larva index, or we call it as angka bebas index, that have been established as the one of the important parameter for the larva survey for the dengue vector surveillance. However, in actually, and the relationship between Aedes abundance and dengue transmission based on the Stegomyia index have some important challenge because of in some countries they have significant relationship. For example, in Havana, Cuba, Trinidad, Vietnam, that reported significant associated between entomological index and the dengue cases. But in some countries, there is no statistically significant relationship. For example, maybe please correct me if I make a mistake, from Malaysia, that published by Sulaiman in 1996, and also in Brazil, Colombia, and some countries, there are reported no significant association between entomological index with the dengue cases. Based on this baseline data, so in our study, in my group, we have been conducted study to identify the correlation between the Stegomyia index and risk of dengue transmission in Indonesia. We use a nationwide survey that conducted in 26 provinces in 78 locations, and we collect entomological data, dengue cases, social demographic data, and also spatial data, and we make analysis together. Sorry. Yeah, this one. And we have been collected more than 65,000 mosquito larvae in a different environmental and socio-economic condition, climate, and also population density across Indonesia, from Sumatra to Papua. And we make analysis to identify the correlationship between the Stegomyia index and the risk of dengue transmission using Kolmogorov mean of normality test and also principal component analysis. And surprisingly, the conclusion is no correlation was found between the incidence of the dengue and the Stegomyia index and free larva index in Indonesia, based on the nationwide data. However, we still use a free larva index, and we know that free larva index is still very useful, especially as a parameter to monitor the success of mosquito breeding habitat eradication in the community, or we call it as to monitor the PSN TBD in Indonesia. Yeah, so I think it's still useful for this. The others dengue vector surveillance method is Ovitrap. Actually, Ovitrap is high sensitive and efficient for detecting Aedes mosquito population at a low densities. Ovitrap also safe, economical, and environmentally friendly tools. And this tool also provide early warning signal of the dengue risk and also cover multiple premises and detect broader mosquito distribution when we compare to larva survey limited to the sample houses. The Ovitrap is also can enhance mosquito surveillance in public area where the larva survey face a challenge due to the lack of household access. However, there are some limitations found when we using the Ovitrap. Ovitrap may produce vector if not maintained properly, and also require a significant manpower, especially for placement and regular maintenance. On the other hand, Ovitrap also, when comparing with the Ovitrap data across different area or over time, it can be misleading because of varying larva habitat or control intervention affect the mosquito oviposition. And the last, this is also some important point. When we found the predation of the larvae in the Ovitrap, the predation by snail or cockroach and disturbance by animal is also can affect Ovitrap data reliability. So this is very common parameter for the Ovitrap. We have been conducted Ovitrap index in some area in Indonesia, and this is the very common parameter and very easy to use. The last of the dengue vector surveillance that also needed to know and very important to implement in the field is adult mosquito trapping. So actually, adult mosquito trapping or adult mosquito collection giving more accurate indicator of the transmission risk than the larva survey, because of it allow a specific species level identification and collection of the mosquito at the different physiological stages. House-seeking behavior, resting, gravid, we can identify by using the adult mosquito collection. And in the adult state, we also use this for virus detection to identify the infection rate of the dengue in the vectors. And we can also conduct blood meal analysis and also insecticide resistance testing. Some strap that we can use for the adult mosquito collection, for example, BG Sentinel trap is also efficient at the collection of seeking female and can monitor a population fluctuation. Adult mosquito trapping also can cover public and peri-domestic area where larva or Ovitrap survey are limited by access or container availability. However, it's similar with the larva survey or Ovitrap. Adult mosquito trapping also have some limitation. Some adult trap collect bias sample due to limit estimate of the overall adult population or infection rate. And then human landing cats, because of in the malaria is very common method to collect the adult mosquito by using human landing cats. But for the dengue vector surveillance, it poses a health risk to collector and requires ethical approval. So I think it's very limited to use these methods to collect adult mosquito in the dengue vector surveillance. High cost logistic and need for regular maintenance is also some challenge in part of the adult mosquito collection, and have seasonal and spatial variability is also required integrity with other surveillance method for a comprehensive risk assessment. Yeah, this is our other study that also have been conducted several years ago to assess of mosquito collection method for the dengue surveillance. We conducted in 39 locations by to assess relative effectiveness of the larva collection, morning resting collection, animal baited trap, and also human landing collection. And this is our finding that after we conducted analysis, the conclusion is no consistency in the efficacy of given method and the incidence of dengue in the human population, and no correlation between any of the parameter considered: method, incidence of dengue, location, and presence of the dengue virus in mosquito. However, we have to maybe choose one or two appropriate surveillance method, especially based on the local specific. Because of in Indonesia, I think this is very important to conduct vector dengue surveillance based on the local specific, because maybe the weather or the location is very different, and also the culture is also different. That it also cause for the differentiation of the breeding habitat in every location. So this is our recent analysis that they have been conducted, and we are preparing for the publication process. Because of this is also important a note, because of after we conducted analysis in 22 districts in 25 provinces in Indonesia. Surprisingly, we collect adult mosquito during whole nights from the 6 p.m. until 6 a.m. in many places. And we also detected some mosquito that have been collected during indoor mosquito collection, and also outdoor positive with the dengue and also chikungunya. So I think it's also need to note, because of actually, even though the most of the Aedes aegypti, also Aedes albopictus, have a diurnal behavior. However, we also found some potential of nocturnal behavior that also have risk for the dengue transmission during the night. So this is also our study in associated to make sure about the population genetic of Aedes aegypti in Indonesia, to strengthening the surveillance of the dengue vector in Indonesia. We conducted in 43 allocation, and this is very good news, because of all of sample belonging to the same maternal lineage, and almost of the sample belong to the same population. So I think this finding is very important, and supporting some vector control project have been conducted in Indonesia based on the population control. For example, maybe Wolbachia, sterile technique insect, something like that is very important to get a correlation between the implementation and all the population of the mosquito. So I think it's good news, and we can conduct it in more area in Indonesia based on this data. So when we back to the dengue control effort in Indonesia, I think this is also important to know, because of the critical strategy for the dengue vector or dengue hemorrhagic fever prevention and control in Indonesia is based on the surveillance system, but in the cases and also vector for planning and response, and disease management, changing behavior and building partnership, and also appropriate vector control. I think the Minister of Health already have very good implementation beside the some complement vector control approach that have been conducted in many places. We have a very strong approach that we call it the source reduction by community participation, by using 3M plus program at the community level and one host, one larva monitor, and combining with some other effective vector control approach. Hopefully it can be have a good result in the future. So based on the strategic national strategic of the dengue control 2021 until 2025, actually We have some approach to control the dengue by especially mainly by one host one larva monitor movement, and also regular larvae monitor by Puskesmas, and multi source collaborative surveillance that just recently conducted in Indonesia and supported by the WHO Indonesia. So this is the one other approach that also important and also have a good good implementation, because of we combine some source to collaborate and combining and to be important to strengthening the surveillance for the especially for the dengue vector surveillance.
I'm sorry, Pak Tri, you have 2 minutes left.
Okay, after this, I think
Thank you.
the last slide. Yes, I think BMKG and also some university and also some district and municipality have been conducted this, and I think this is a good news for us. So for the conclusion for this, my presentation today is overall a multi-method surveillance approaches combining a larva survey, ovid trap, and adult mosquito collection is critical for effective dengue vector monitoring and control. Traditional Stegomyia index and free larva index, actually, even though there is no consistent correlation with the dengue incident, however, we still need it, especially to monitor the larva source control in the community. And ovid trap are highly sensitive, efficient, and economical tools for detection Aedes mosquito population, especially at the low densities. And despite this advantage, so ovid trap require proper maintenance to avoid inadvertently production of mosquito and their deployment in a resources intensive. The last is adult mosquito trapping method provide more accurate indication of the transmission risk than larva survey, but have challenges including cost, logistic, and potential sample biases. Thank you very much, Professor Eggi. Yeah, this is
Thank
my
you. Thank you, Pak Tri. Please stay with us
okay.
because I believe many questions will be addressed to
Okay.
you after this. Okay, so we move to the second speaker for this session. Yeah, today we are very grateful that today we'll have Associate Professor Lucas Low. I'm sorry, can you please back to the CV? Okay, so we are very grateful to have Associate Professor Lu Van Len from TIDREC University Malaya. He will talk about the novel vector control technologies, and he is the Associate Professor and Executive Director at Tropical Infectious Diseases Research and Education Center, or TIDREC, University Malaya, Malaysia, and also President of Malaysian Society of Parasitology and Tropical Medicine, Associate Editor of Tropical Biomedicine Journal, and also one of his research as the principal investigator of the Wellcome Trust grant, the Tick Cell Biobank, an arthropod vector cell lines for human and animal health. So, Associate Professor, time is yours.
Thank you, Prof. Arguni, for your kind introductions. Let me share my slide. Can you see my slide?
Yes, clearly.
Okay. Okay. Thank you very much for the opportunity for me to be here today to share a bit of my research on the use of the Streptomyces bacteria, secondary metabolite for mosquito control. So today I will be very much focusing on the molecular aspect of these Streptomyces mechanisms towards different mosquito species. And specifically, we try to identify the possible mode of action of this novel bioinsecticide for mosquito control. a little bit information about Streptomyces bacteria. So this is a high GC gram-positive environmental bacteria. And this Streptomyces is well known with various activities. These include the antibiotics, antiviral, antifungal, antiparasitic, and even anti-tumor, and also other activities as well. So the products of Streptomyces include the well-known product antibiotic streptomycin and also tetracycline, as well as the antiparasitic drug ivermectin. The use of the Streptomyces-derived compound for the control of agricultural pests have been well documented. These include the moth, the flies, and also some species of the ticks. But the studies on the mosquitoes have been relatively understudied. And accordingly, today I'm going to introduce Streptomyces KSF103 strain, which was isolated from the soil samples from Pahang, Malaysia. And this strain has the highest similarity with the Streptomyces rubisoli, which was originally described from China. So these are the three ladies that have contributed a lot to this project. The first lady is Dr. Aziz, who is specialized in Streptomyces and natural product. And the other two ladies are the master student and also the PhD student for this project. Both of them have been graduated. And today I'm going to share with you the assessment of toxicity of Streptomyces KSF103 secondary metabolites against the mosquitoes, and also as far as the environmental friendly is concerned, we also test on the non-target organisms. And we attempted to identify the possible mode of actions of this bioinsecticide against the mosquito cell line and also the mosquito of different species. And here is the overview of the toxicity assessment. We obtain the bacterial cultures and do the ethyl acetic extractions, and we dissolve in the DMSO always for the larva bioassay and acetone for the adult bioassays. And this toxicity assessment we further divide into two different set of experiment. The first set, we perform the cytotoxicity test to test on the C636, which is the Aedes albopictus cell line, and also the non-target human lung cell line, MRC5. And on the separate experiment, we perform the toxicity test on the eggs and also the larvae, as well as the adults of different mosquito species. And we also perform non-target organism bioassay, which include both marine and freshwater macroalgae, as well as the ants. And these show the results of the cytotoxicities of Streptomyces KSF103 against the Aedes albopictus cell line. And the EA extract, the ethyl acetic extract, has some considerable level of the toxicity towards the cell, and hence it was selected further for the insecticidal testing in the mosquitoes. And whereas for the cytotoxicity effect of this bioinsecticide extract on the MRC5 human cell line, it displays some toxicity effect, but it requires much higher concentrations, so about 10 times higher concentration needed for the target cell line, which is C636 Aedes albopictus cell line. And here is the results for bioassay against different life stages of the mosquitoes. This include the larvae, the adult, and also the eggs of different mosquito species, including two major dengue vector, Aedes aegypti, Aedes albopictus, and also the simian malaria vectors, Anopheles crescens, as well as the most nuisance night biter in the residential area, Culex quincke fasciatus. And here we show the range of the LC50 and also the LD50 that we have obtained from the experiments across different life stages. And the Streptomyces KSF103 display no toxicities on the freshwater as well as the marine microalgae as well as the ants. So suggesting that they are environmental friendly. And given the promising results from the bioassay part, so we proceed with our second objective to identify the preliminary mode of actions of this bioinsecticide against Aedes aegypti as a study model. So we also perform two separate tests. One is on the cell, C636 cell, followed by the cytotoxicity test as well as the flow cytometry assay. So this flow cytometry assay is a tool used to detect the presence or the evidence of apoptosis after the exposures of the insecticides. So, and we also performed the bioassay as well using the LC50 or the LD50 of the larvae and adult stages. We expose them with these sub-liter concentrations, and we used the exposed mosquito and also the untreated mosquito as well for the subsequent work. This includes the biochemical assay, which we target different enzymes that are known to be involved in the detoxification mechanisms. And also we want to identify the up and down regulated genes after the exposures of this insecticide. We perform RNA analysis through the microarray analysis. And once we have identified the genes down or up or down regulated, so we further validate our finding using the gene expression study, qRT-PCR, based on our newly designed primers. And this show the results of the flow cytometry. And as expected, the insecticide show the evidence of the apoptosis on the Aedes albopictus cell line, depending on the doses that we use. So it is on the dose-dependent manner. The higher the concentration used, the higher apoptosis rate detected in this study. And another aspect, we performed the biochemical assay targeting different enzymes, including acetylcholine esterase, alpha esterases, beta esterases, and mixed function oxidases, as well as the glutathione S transferases. So among these 5 tested enzymes, only acetylcholine esterase show the significant correlation between the treated and untreated, and show this association, which suggests the toxicity may have disrupted the insect nervous system. And for the microarray analysis, we have identified the top 10 genes with the highest apoptosis change. There are many genes detected, but we only focus on the top 10 genes. This is the genes that we have identified at the larva stage, and they are up and down regulated genes. And this is for the adults, mostly up regulated. And to further verify our finding, we need to find the genes that are commonly detected across adult and also larva stages. And here we identified 2 genes that showing up regulations after the exposures. So this include the tropomodulin genes and also the cestrin homolog genes that have expressed across both life stages. And to further confirm our finding from the microarray analysis, we use a qRT-PCR to validate the genes. And indeed, we found the significant expression of cestrin and tropomodulin genes at both larva and also the adult stages. So this confirms our previous finding on the microarray analysis. And we further perform functional analysis based on the genes that we have identified from the microarray analysis. And here we show two signaling pathway that may be involved in the apoptosis. And this includes the MAPK and also the Notch signaling pathway. Both pathway have been associated with apoptosis. And now we look at another aspect. So we have proved the evidence, the molecular evidence. Now we want to look at the proteomic aspect as well. And we perform the LC-MS analysis on both treated and untreated mosquitoes. And here we proceed with the shotgun protein identifications analysis. And these are the three genes that we have identified at both larva and adult stages. The three proteins, not genes. Yeah? These include the ATP synthase, which has been known to activate the defense mechanisms via the energy productions, and also the FPA, fructose biphosphate aldolase. This also has been known to be involved in the oxidase stress by the insecticide. And last but not least, the ATP citrate synthase that also involved in the Krebs cycle. This is also important in increasing the mitochondrial activities, energy production, as well as the detoxification of the insecticides. So here are the three genes that are associated with insecticide exposures and detoxifications. And someone may argue that we are using extract, so EA extract or any other form of the extractions. They are not in the purest form, so we are not using the pure compound. So we can't really identify which compound actually playing the insecticidal activities. And we acknowledge the limitations, and here we perform the LC-MS analysis as well on the EA extracts. And here we have identified several compounds, and we perform the molecular docking with the proteins that we have identified from the mosquitoes. So the protein of the mosquitoes versus the compound from the EA extract. And here we found the FPA may serve as the target, the protein target for these insecticidal activities, given the high binding affinity with two compounds. This is dodimorph and also the cellagines. And as the conclusions, the present study has identified that Streptomyces KSF-103 EA extract exhibited in vitro insecticidal effect on the C636 albopictus cell line. And this extract also show promising results on the larvicidal, adult decidal, as well as the ovicidal effect on different species and different genus of the mosquitoes. And only minimum toxicity was observed on the non-target human cell line, as well as the Cholera, macroalgae, and also the ants. And the flow cytometry showed the evidence of the apoptosis. And the biochemical assay show acetylcholine oxidase inhibitions. So which we suggest the toxicity may have disrupted the insect nervous systems. And tropomodulin and cestrin demonstrated the involvement is in the preliminary mode of actions of these extracts, which involve the apoptosis induction in the mosquitoes. And the functional analysis review association with the MAPK and also the Notch signaling pathway, which are responsible for the apoptosis as well. And last, the FPA may serve as the potential target protein for the insecticidal activities of these EA extracts. And before I end my presentations, just a small announcement regarding the upcoming event organized by Malaysian Society of Parasitology and Tropical Medicines for its annual scientific conference. So this conference is to be held in 30th and 31st March 2026 in Ipoh, Perak, which is around 3-hour driving distance from Kuala Lumpur. And here are the list of the speakers that have confirmed their availability. And if you are interested with this event, please scan the QR code here for more information, or you may contact us through the msptm.event@gmail.com. So with that, I end my presentation. Thank you very much.
Thank you, Dr. Lu Van Loon. It is very interesting and very intriguing presentation because for me, for example, like now currently we just know about the Wolbachia as the innovative vector control to prevent dengue infection, but today we heard about the new one, Streptomyces species, and then I believe many questions will be addressed to you after this session or during our Q&A session. Thank you. Please stay with us. Okay, so we move to the third speaker, Dr. Citra Indriani. She will deliver the topic of pilot implementation of Wolbachia technology in 5 cities in Indonesia. She is now the director of the Center for Tropical Medicine, Faculty of Medicine, Public Health and Nursing, Universitas Gadjah Mada. Also lecturer at the Department of Biostatistics and Epidemiology and Population Health, and currently acting as a co-investigator and epidemiologist in the implementation of Wolbachia technology in 5 cities in Indonesia. So, Ibu Citra, give the time for you.
Okay, Prof. Eggi, thank you for the kind introduction. I would like to share on the slide first and check it. Yeah, is it work? Is it?
Yes, perfectly.
Okay, yeah. So, good morning, everyone. First of all, I would like to thanks for the organizing and also Bu Eggi for initiating this grateful webinar series with Universiti Malaya, also Bu Inge from Universitas Sumatera Utara. Yeah, hopefully the next webinar will be also as great as this year's Bu Eggi for commemorating the World Mosquito Days. Yeah, so the organizing committee is asking me to present related to the Wolbachia pilot implementation. This slide actually built by Dr. Iris Andono Ahmad, who is the principal investigator for this pilot implementation in the Center of Tropical Medicine, and also Prof. Eggi also as the co-PI for the project. So this is my outline. The first of all, would like to share the dengue burden in Indonesia, and the next is to the Wolbachia replacement methods, and then we go to the research and implementation of WRT, and the last one is discussing about pilot implementation. So if you look at this picture, we can see in here the burden of dengue in Indonesia has a similar situation as the Dr. Ajib, our previous speaker, is mentioned. Yeah, it's bigger and bigger, and we realize that there is a expanding risk and also expanding threat also related to the dengue. The last 2 decades, we can also see the exponential pattern that is notified in here, and with the initiative and also program implementation since 70s up to now, the burden is still exists, the threat still growing up. Despite the initiative and the innovation, based on this we merely need an innovation, and one of the innovation that currently seen as a promising method is and on 2022 the MOH now is adopted the Wolbachia as part of the pilot implementation project in 5 cities. So discussing about Wolbachia, Wolbachia is an endosymbiont bacteria and it is naturally found in more than 50% of insects. The unique of Wolbachia, if it is exists in the Aedes aegypti, it has a unique mechanism that we call as a cytoplasmic incompatibility. And what is the cytoplasmic incompatibility is when the male Wolbachia meet with the female one, Mosquito is not producing the eggs, and the eggs actually produce the egg, but the egg is not hatching. So we call it as no viable offspring. And then if female Wolbachia meet with the male one without Wolbachia or the wild type, so all of the Wolbachia, all of the eggs is viable and containing Wolbachia. And also if both are Wolbachia, all of the offspring contain the Wolbachia and the eggs also viable. And then the other things, when Wolbachia is exists in the mosquito, the Aedes aegypti mosquito, it's also reduce the capability or the competence vector in transmitting the dengue by blocking the viral replication. And the Wolbachia replacement is actually use the this benefit of Wolbachia infected Aedes aegypti that can be used to reduce the vector competences also to what you call it? We need to spread the Wolbachia across the mosquito population, and we use the cytoplasmic incompatibility. So the Wolbachia replacement therapy method is using the male and female. Then we also know that there are another method, we call it as suppression. They use the potency of male mosquito for suppressing the population. And the research, Wolbachia replacement technology on research and implementation, we started or initiated in Yogyakarta since 2011 with collaboration with National University and World Mosquito Program, and it funded by the Tahia Foundation. It's a national NGO in Indonesia. Started with the feasibility up to the efficacy study. The efficacy study, it designed it as a trial study, and the result we already get the result. The result is we found that Wolbachia has a 7.1% of the efficacy. The researcher commitment and also their persistent advocacy continue the result of the research not only on the paper, but then they are doing the advocacy to the national level and then also in the global level at the WHO together with the collaborators. But then there's that the research method is it not applicable to be used in the program model implementation. So they continue to do the implementation research for finding the model for programmatic implementation. And then, yeah, so we already published a document also that documenting the model, which we developed in 2022. And then in 2022, also national or the Ministry of Health then adopt the Wolbachia into it's a program as a pilot implementation in August 2022. Yeah. So this is the, after the trial, this is the impact on dengue incidence beyond the trial, or the trial is called the AWE trial. We can see in here before the Wolbachia period, we can see amplitude and also the wave of dengue in monthly incidence is high, and also the wave is. But then after the Wolbachia implementation, we can see that the reduction is notified during almost 20% of the coverage area. But then after the whole city are covered by the Wolbachia, so we see the sustained reduction on the dengue incidence, on the monthly dengue incidence compared to the previous era when the Wolbachia has not been implemented. So, but then Wolbachia, if we can see in here, Wolbachia is not eliminate the dengue at all in here, but we can see the reduction in here. And then we also seen the other benefit of Wolbachia. We see fogging, the number of fogging that needed to do the response for outbreak. For the dengue outbreak, we see in here the blue bar indicates the number of fogging events in the intervention area, and the gray bar is the number of fogging events that occur in the non-intervention area. We can see after the Wolbachia implementation, we see the significant reduction in the area. And this reduction is also give a benefit. Some of the budget allocated for the fogging is shifting to the other program for the strengthening other program implementation. So after Yogyakarta, we go to the 5 cities Wolbachia implementation. It starts on 2022 for the planning, but then the implementation is starting on 2023. So before we move forward on how Wolbachia is deployed, I would like to give you a brief introduction related to the Indonesia health system. So we have a national level is the Ministry of Health, and then there is a health office in the provincial level, and then also district health office in the district level. And the district level is coordinated or many sub-district public health center. So the first responder on the dengue control is who work in the puskesmas, and also those who are working in the district level. And because of the autonomy era, each district level has their own budget also. So we called it as a local budget. And the puskes also, they act as a unit service, so they can earn some money for giving a service, but also have some from the district level and also national level to do the programmatic for the public health prevention program. So in Wolbachia replacement method implementation, we need 5 components that we identified during the implementation research on 2021. So the 5 components consist of the organization and management. The second is communication and community engagement. The third is partnership. The fourth is the deployment strategy, and the last one is monitoring evaluation. So these 5 components needs to be access for implementing WRT. And our deployment strategy, we use the X in the ARC or in the bucket, and we deliver it bi-weekly. And the deployment is last for 6 up to 8 months. We use the X and strips, and currently also use X in capsule. So there are 2 package for the X. In each 5 cities, they have their own program and they have the name of their own program. For example, Winko in Semarang and WIS in Bontang. And this also express their ownership for the Wolbachia implementation integrated to the dengue control program. But then 4 cities are during the implementation in the initial on 2020, 4 cities are heavily affected from this information that resulted from the Bali implementation. This is the characteristic. The Semarang has the biggest area, and we have the ERC grid. The grid size is 50 meters multiplied by 50 meters. And then we use the release cycles for about in approximated 10 cycles. So in each cycle, we need 2 weeks. So I mentioned before, the release is bi-weekly. So this characteristic for the implementation area, and then this is the organization structure. We have in here UGM, our center as technical assistant team, and also producing the Wolbachia IDS HFDX. And other than UGM, we have the production unit from the MOH unit in the Salatiga. And also currently in this year, we also have support by Udayana Insectarium that support by Wood Mosquito program. The coordinating and also the budgeting coming from national and also on the local in the district level, they lead the program, and also our team, the technical expertise and also the field team, they work closely in the village and also working closely with the DHO, also led by the dengue program manager at the district level. And we also include the public health laboratory monitoring and also laboratory at the port health for identifying mosquito during the Wolbachia monitoring. This is the budget resource. We use the national budget and also the local budget. And also this year we have support from the DFED in the strengthening the implementation. This is the process how we implement the plan. So the first process is to conduct the mapping. We need to map the area that targeted for the release in a grid with the size 50 multiplied by 50 meters. And then after doing the mapping, we continue to the community engagement, doing the dissemination, the socialization related to the Wolbachia, and also to gain some insight of their asset. And then we, because we use the ERC or egg release container, we need the host for the ERC. So in here, we put the host code identification. We identify the potential host that wants to support the program. And then we do the first release, or usually we mention as a launching. And then after the first release, we did the ERC service. It means that we replace the water and also put the fresh egg again in the ERC. And then after 6 up to 8 months, we withdraw all of the ERC and we say that all of the deployment is completed. During the release, we do for monitoring Wolbachia, and after the withdrawal of the ERC packet, we do 2 monitoring. And monitoring, this is the process from preparation and then mosquito sampling, and then we use the sweep net, most of the area using the net, but then if in the area that we cannot do the sweep net because difficulties on finding the house or the permission of the house owner, we do the PG trapping. After the sampling, we send it to the port health laboratory for identification and preservation, and then ship it into the port health, into the public health laboratory under the MOH to do the Wolbachia screening. And then all of the screening process, part of the monitoring and evaluation, we also conduct the verification or the quality assurance from the testing sample. And for Kota Semarang, one of the biggest city that we deploy, we can see in here almost of the Semarang area, about 30% actually, because this is a sub-district level, but actually not all of the sub-district level has the implementation. Because scatter area based on village level, so we put in here as a kecamatan level monitoring result. We can see here some of the area has reached obtained the targeted Wolbachia frequency, which is 60%. Then still some areas still in process and haven't obtained the targeted Wolbachia frequency. In the phase one, it's already completed, and the four sub-district in the below map. But then we have yes when the disinformation is coming up, so it hits a lot of the participation. So many participants is withdraw during the implementation. And also we face some challenges on the problems, but then we learn from the previous phase, and then we have optimized the production and also have a close monitoring in the production. And this is the current monitor of the dengue in Kota Semarang. The green line is is for the intervention area, and the red one is for the non-intervention area. The green shading in here is indicated the Wolbachia deployment period, and we can see in here, because actually after the big hit in 2024, we usually see in many areas also the decrease dengue cases. So for our side, actually, maybe there is an indication of a good Wolbachia impact on the dengue cases, but to see confidence to the impact, we need to be patient. So we need to wait until the next dengue season in Semarang City.
Mbak Citra, you have 2 minutes left.
Yeah. Okay.
Thank you.
So for the Wolbachia frequency, all of the area have been successfully implemented, and the Wolbachia frequency is obtaining during the implementation. But in Pontang, we see after the implementation is going down. But we are planning to see more. To see the Wolbachia frequency in next October to know is it going to decrease, then we have a good result on Wolbachia. And then I will skip this. So this is the challenges that we face during the implementation. We have a fragmented budget from the national level and the local level, and also fragmented decision making. It makes the implementation is challenging. And also the rigidity of regulation and financing allocation, and the use makes the money is not flexible to be used. And also we face the local conflict of interest, balancing between the resource availability and also the local political leaders' expectations. So it also raised the challenges. And also on the local capacity in the leadership, the area without the leadership, so the implementation also lose less monitoring, and it is also tough for the Wolbachia establishment. Time for funding disbursement at the different level and budget allocation, and also time of the planning of the implementation also been a challenge for our implementation. So in the summary, in the phase one, Wolbachia implementation is completed successfully in 4 cities, and Wolbachia Mosquito were established in all implemented area. However, 2 sub-districts in Pontang require further monitoring because of low frequency after deployment. The implementation process was challenging due to the fragmented structure and system required for ideal implementation. I think, yeah. So thank you for all the people of Yogyakarta and all the places when the plan was put into an. And thank you for the organizing committee and kind participation from the participant.
Thank you, Bu Citra. Yes, it is very promising still, yeah, because we are not finished the monitoring yet for all of 5 cities. But the result is have been waiting for a long time to everybody. So it's very nice to have the result later. Okay, so we move to the Q&A session. Yep. Perfect. So, question to Bapak Triwibowo, if the traditional pneumonia indices show no consistent correlation with dengue incidence or transmission risk, then what tools can we use so that we have representative result on preparing for dengue outbreak? Yeah.
Okay, thank you very much, Prof. Eggi. Thank you very much for the question from the audience. So I think yes, that's right because of now we have beside the Sp.A index, I think now we have some promising methods to propose as the predictor of the dengue risk of transmission, especially it's come from the Malaysia because of Professor Indra Fitilingam and team. I read a very nice paper from University of Malaya. They already have one of the solution that we also adopted in Indonesia, and now we in a preliminary study for this. We're using dengue virus based for the entomological study. We're using NS1 strip to conduct the entomological study. So we collect adult mosquito in the field, and then after we collect by using sweep net or aspirator or maybe a BG sentinel trap, then the mosquitoes surrounding the area we brushing together, and then we put PBS1, and then we drop it in the NS1. If the positive, it will be a very important information because of the current situation is the dengue transmission ongoing in this area. So this is a good one, and I think we will continue for our study to adopt this finding. And I think the others promising method is come from the BMKG, from the Meteorological Institute of Indonesia, and also in collaboration with the Jakarta provinces. They also conducting I think a very good analysis to predict by using climate prediction. So I think now this activities are already ongoing, and they have a good result regarding this. So I think maybe a multi-sectoral collaboration for the surveillance needed to discuss this, and then I think hopefully we can get the best one for the prediction for the risk of the dengue transmission. I think before that, I think we can still using Sp.A index, but maybe in the future, I think we need to better implementation for the predictor for the dengue risk of transmission. Thank you, Professor Eggi. Yeah.
Yes, very good. I hope Bu Mirsa can accept that explanation. So move to the second question to Dr. Lucas. What is the future direction for the use of streptomyces as larva control method? Whether the study will be expanded to bigger project to finally reach product development phase, as your presentation mentioned, promising result. Please, Dr. Panlu.
Thank you very much for the question. For now, I think it is a bit difficult to proceed with commercializations because, as I mentioned, it is not at the purest form. So it is a mixture of different compounds So we can't really identify which compound that actually playing the insecticide activities. So, but I think it does not limit us from moving forward. But what we can do is that we can actually do some control that can complement the existing vector control strategies. Like this one, we can do the sugar beta trap infused with this streptomyces extract. Or to move forward, if we want to aim for the commercializations, we would need to do the purest form of the compound. So which we are now also planning to do that. So to test on the pure compound instead. Then from there we can move forward.
Okay. Because it is also my big question, I may ask the committee to allow me to ask this first to Dr. Finland. It is I mean, the streptomyces is exactly like a bacteria, right? So in your study, what is I mean, what is the side effect or um, I mean, if we have this kind of factor control, so we want to spread it to our environment, if I'm not mistaken, like that. And then what is the side effect of this bacterial in our environment?
I think I need to clarify. We are using the secondary metabolite, so we are not using the live bacteria to
I
be
see.
released into the environment. So I
Sure.
think the effect would be the same as the conventional insecticide. They may have the toxicity against other non-target organisms, but in our study we only tested 3 organisms: the microalgae and also the ants. But we would not know what's the effect on others insects. But looking at the target organism, specifically for the mosquitoes, they are so tiny as compared to the butterfly or dragonflies. So I think the toxicity would be very minimal.
Ah, yes, I see. Thank you for the explanation. So can we move to the next question? Okay. To Ibu Citra, climate change is causing an increase in the mosquito population. Does it also happen to Wolbachia carrying mosquitoes?
Yeah, thank you, Ibu. So I think because it also affected the uh, population overall. So if the area is already implemented the Wolbachia infected mosquito, so I think it will be say the similar. It's also will be affected with the climate change when the uh, more rainfall, more breeding sites, so then the population will increase. But I think Pak Garjito could add more to the answer.
Yeah, Pak Tri.
Okay,
Y-
yes,
y-
it's about the weather, right? With excuse me, maybe Professor Eggi, can you open the question? I will continue
Yeah,
the,
yeah.
yeah.
Can we see the question?
Not yet, not
Yeah.
yet,
Okay.
not yet. Yes. Okay.
Yes.
Oh, yes, yes, yes. Actually, this is maybe a different discussion when we talk about the climate change and also Wolbachia carrying mosquito. Actually, so far we have information and we have some evidence in associated with the faster replying of the virus in the mosquito body and also for the more the longer lifespan of the mosquito during the climate change. So I think maybe it will be give a good result maybe in the future, or maybe we need to make some further study for this, especially in association with the Wolbachia, because of maybe if the mosquito with Wolbachia they have a longer lifespan, I think it will be good also in the community, in the environment, because it also give the stronger spot for the to protection the transmission of the virus to the human. This is maybe we still challenging because of there is no evidence about this study about this. But maybe for the other situation, if for the faster replication of the virus in the body of the mosquito, and maybe it will be also continuing with the faster replication of Wolbachia also in the body. So I think it's really challenging, and I think hopefully it gives the positive, not the negative issues for the Wolbachia, because of so far we just worry about the spreading of the mosquito to the wider area in the world due to the climate change. But for the others, I think we need to further study about it. Yeah.
Yes, I agree, Pak Tri, and also Ibu Citra, because yeah, if not mistaken, that the for Wolbachia mosquito, they do not have any changes in the structural and also functional, right? Only for several strain, they can change the functional, but the strain that we use in Indonesia, until now, I think we have the evidence that the structural and also functional of the mosquito is not differ from the other mosquito. Okay, very good. Yes, do we have another questions? The committee, can you share the questions, or maybe I can open it from the
Yeah,
chat
Prof.
box?
Eggi, there is no more questions, I believe.
Okay. Oh, okay. Good. I think several questions still unanswered. So I hope Dr. Citra, Pak Tri, and also Dr. Van Loon can see the question and also can answer it privately. Thank you so much. And I'm very happy that today we learn so much about the factor control. We know that our indices or our parameters for factor surveillance is not ideal yet until now, but still we can continue the factor surveillance using that indices. But we believe that several innovative parameter can be used as well. And also the new factor control already explained by Dr. Van Loon, that it is also promising, but I still believe that for dengue we do not have one silver bullet. So we have to do all of this strategy, not only Wolbachia, not only this streptomyces factor control, not only vaccine, yeah, but also but all of the strategy we have to do it integrating. So thank you so much for the speakers of this second session.